Hello and welcome again to the next topic, 1.2, Students T-Test and Correlation. I hope you enjoyed the last video, and this is the last video that we are going to do about statistics, so please don't be disappointed if what the topics here are very boring because it is math or because it is statistics, but this stuff is really necessary and you will find it very useful, especially when you are writing your lab reports and using the data that you have gotten in the lab yourselves. Okay, so part 2 for statistics involves the student's t-test first, and then we are going to be talking about correlation between two variables. Okay, so to get started, this t-test is a statistical tool, a statistical test that you can use, and it's called student because the person who first came up with it, who was William S. Gossett, who was a person that was working at the Guinness Brewery in Ireland, he first published it anonymously under the name student because his employers did not want people to know that William Gossett was using statistics for the Guinness Brewery. So if you are interested into this story, it's kind of neat. You can Google it. You can look it up on any resource on the internet. Okay, so why would we want to use a student's t-test? Brings us back to the question that we asked in our last video. Are our results reliable enough to support a conclusion? That means you have calculated your data, you have taken the average, for example, of your data set, you are comparing two populations or whatever, and you want to know without a doubt whether your results support a conclusion. Can you say that the hummingbirds are significantly different or not, and so on. Okay? So in this case, we are going to imagine that we choose two children at random from two classrooms. Okay, classroom D8 and classroom C1. So we're just going to choose one student from each class, and we are going to compare their height, how tall they are. Okay, it turns out that we find that one pupil is taller than the other. Okay, the pupil from C1 is taller than the pupil from D8. Now we ask ourselves, why? Well, there may be many reasons. Reason number one can be that there is a significant difference between the two groups. That means that the pupils in classroom C1 are generally taller than pupils in class D8. Okay? Reason, for example, class D8 may be 7 years old, and class C1 may be 11 years old. So that would mean that class C1 would be taller in general than class D8. Okay? Another reason for this, it could be that by chance, by coincidence, we picked a short pupil from class D8 and a tall one from class C1. Maybe we chose Titch, who is 9 years old, from class D8, and maybe we chose Hagrid from class C1, who is also 9 years old, but it turns out that he is taller than Titch. Okay? So how do we decide which reason is most likely, reason number one or reason number two? So in order to decide, we need to measure more students. If there is a significant difference between these two groups, okay, we measure more students, we can see that a lot of the students from class C8 are going to be short, and a lot of the students from class of C1, the average of these classes, or the mean height of the two groups, should be very different. Okay, because in general, all the students are different. However, if there is no significant difference between the two groups, okay, class D8 and class C1, in this case, the average or mean height of the two groups should be very similar. Okay, so the first thing that you need to remember is that living things normally show a lot of variation. Remember that you're talking about living human beings that are in the process of growing, especially if you're talking about students in a classroom. They may be one height one day and a different height the next day, or they may have finished growing, but it's something that is so uncertain that there is a lot of variation naturally in biological things. So it is very unlikely that the mean height of our two samples will be exactly the same, but they are going to be somewhat close. Okay, for example, we have our C1 sample. You see the students there, and the average height is 1 meter and 62 centimeters. Okay, in class D8, the average height is 1 meter and 62 centimeters. So is really 162 very different from 168? Is it significantly different? Can we say that 6 centimeters is a lot of difference? Depending on what we are looking at. Okay? Is this difference in average height of the samples large enough to be significant? How can we tell for sure? Some people, you may ask, does 6 centimeters make a difference? Well, maybe not if you're talking about choosing the type of chair that they're going to sit in. Okay, 6 centimeters may not make a lot of difference. But if you're choosing, for example, pants for their uniforms, 6 centimeters may be a large difference. Okay? So, we can always analyze the heights of the students by doing a histogram, which we talked about in the last video. Okay, so here are our histograms for class C1 and for class D8, and you can see that we are graphing frequency on the y-axis, and on the x-axis we have their height. That means that if you look at the top C1 sample in yellow, if you look at the first bar of the histogram, it means that 7 students measure between 140 and 149 centimeters. Okay, whereas 2 students measure between 160 and 169. Okay, in the D8 sample case, the first column, the first bar, it says that 1 student measures between 160 and 169 centimeters, and then the other students measure more than that. Okay, so here, if by looking at the histograms here, the ranges of the two samples have a small overlap. That means that they are, if we were able to put one on top of the other, they would not coincide very much. They would be very different. And so the difference between the means of the two samples is probably significant. Okay, remember this is what we talked about in the last video. In this case, if this were the case of the sample in C1 and in D8, here the ranges of the two samples have a large overlap. So in this case, the difference between the two samples may not be significant because a lot of the data is shared. Okay? The difference in the means is possibly due to random sampling error. It might be that we chose just randomly, we chose the shorter ones from one class and the taller ones from the other class. Okay, so in order to decide if there is a significant difference between these two samples, we have to compare the mean height for each of the samples. And we also need to compare the spread of the heights in each sample. That means that we need to use what we talked about last time, the standard deviation of a sample, as a measure of the spread. Okay, remember this formula, we talked about it before. You can calculate the standard deviation using this formula where S is the standard deviation, X stands for the individual measurement of the sample, that means each student, N is the number of samples, and by calculating the standard deviation of a sample, we can also use the t-test, because the t-test is going to compare the averages that we have calculated from each classroom and the standard deviation for each classroom and see if there is a significant difference between them. Okay? So in order to start calculating, or in order to start using the t-test, we have to calculate a number that in this case is going to be called t. Okay? So t can be calculated using the following equation. It's very similar to the standard deviation equation because it is comparing the means and the standard deviations. Okay? If you look at this formula, you can see that the x1 and x sub 1 and x sub 2 with the bars on top represent the mean of sample 1, the average of classroom C1, minus the average of classroom D8, okay, and that number divided by the squared standard deviation for each of the classrooms divided by the number of students in each of the classrooms. Okay, so we're going to work an example with this formula so that you understand it. We're going to go step by step. The t-test tells us the probability of two data sets being the same. Okay, we are going to talk about, we're not going to go in too much deepness about what the t-test does and what the p-value means and what the critical values are, because that's a little higher statistics that we may talk about when we talk about ecology. We may talk about statistics again, but for now, for this introductory section about statistics, we're not going to worry too much about this. But in this case, the t-test has another value that is called p. If p equals 1, that means that the two sets of data are exactly the same. As you can see in this graph at the bottom left, you can see p equals 1. That means that the data are identical. Okay? If p equals 0, the two sets of data are not at all the same. They are completely different, as you can see on the right-hand side. In the graph in the middle, you have p equals 0.5. That means that half of the data sets are the same and half of them are different. So the closer we are to 0, that means that the more different the data are, and the closer we are to 1, that means the more overlap there is. Okay? All right, so in order to use the t-test, we calculated the t-value, and in order to use the t-test, we always start by stating the null hypothesis. Okay? In this case, the null hypothesis is there is no significant difference between these two samples. Okay, so we start by using the null hypothesis, and our null hypothesis in this case is going to be that the students from class C1 and the students from class D8 are not significantly different. Okay, and the null hypothesis is always the same. Whenever you're using the t-test, you are always going to use this null hypothesis that there is no significant difference. Okay, if the value for t, whatever we calculated for t, if our t-test instructs us to accept the null hypothesis, we are going to see how we can accept the null hypothesis. It means that the two population means are not significantly different. We are accepting the null hypothesis. There is no significant difference. Okay? If the value of t instead instructs us to reject the null hypothesis, then we can say that there is a significant difference because we are rejecting the null hypothesis that says that there is no significant difference. Therefore, there is a significant difference. Okay? If that doesn't make sense, pause right here very quickly and read this over slowly until it makes sense. Okay? But it is quite simple. Okay, moving on. Let's look at this worked example. Okay, here we are taking random samples from pupils of classes C1 and D8, and we have recorded their heights. You can see the heights in centimeters of students in C1 and of students in D8. Okay, in this case, step number 1 is going to be to work out the mean height for each sample. So we calculate the average of class C1, which in this case is 161.6 centimeters, and the average for D8 is going to be 168.3 centimeters. Okay? Step number 2 is going to be to work out the difference between the means. So we need to subtract one from the other. Okay? And our value for that is 6.67. Step number 3, we need to work out the standard deviation for each sample using either our calculators or Excel, or we can do it by hand using the formula if you want, but the standard deviation for C1 is 10.86 and the standard deviation for D8 is 11.74. Okay, so step 4 is to calculate the square standard deviation divided by the number of students for each sample. For class C1, this value is going to be 10.86 squared divided by 15, and that's going to give us a number 7.86. For class D8, it's going to be the same thing. It's going to be 11.74 squared divided by 15, and that's going to be 9.19. You can always check these numbers in your calculator, follow these calculations to make sure that you know how to use your calculator first of all, and then to make sure that I am doing the calculations correctly. Okay? So step number 5 is going to be to work out the square root of all of that we've calculated. Okay? That is going to be 7.86 plus 9.19. The square root of that is going to be 4.13. And step 6 is going to be to calculate t. Remember, the formula is going to be this, okay? And that's going to be step 2, that we calculated the difference between the means, and we need to divide that by what we just calculated in step number 5. So that's going to be 6.67 divided by 4.13, and that's going to give us a number 1.62. We can calculate this value t for a pair of data sets and compare it to a calculated critical values dependent on a number of predetermined factors. That means that a lot of calculations have been made depending on different conditions, different critical values, that we'll talk about it in a little bit, and a table has been made. This is called the critical values table or t-test table, and we can use that table to compare the number that we got for t. How does it compare to a critical value? That comparison is going to allow us to either accept our null hypothesis or reject our null hypothesis. Okay? So this table you will see in the next slide, and it is called the t-test table. But step number 7, in order to use this table, you need to work out the number of degrees of freedom. And what degrees of freedom is, you don't need to know, but you need to know that it's based on the number of samples you have measured. That means you need the degrees of freedom is the total number of samples minus 2. Okay? Here we have 15 students from class C1 plus 15 students from class D8 minus 2. That's going to give us the number 28. So our degrees of freedom is going to be 28. We can say that we have 28 degrees of freedom. Okay? Right now you don't need to know what exactly that means. All you need to know is that you need to calculate the degrees of freedom in order to use the table that I am about to show you. Okay? Step number 8 is going to be to find the critical value of t for the relevant number of degrees of freedom. Again, what exactly this critical value is, you don't need to know. But what you need to do is find the critical value for 28 degrees of freedom. Okay? For biology, always use the 95% confidence limit. That means a value of p equals to 0.05. This is what it means. Okay, when you look up on the internet a t-test table and so on, you are going to find a table that looks like this, where on the y-axis or the first column we're going to see the degrees of freedom. Okay, and then at the top row you are going to see the values for p. Remember that the smaller the value for p, the higher confidence there is. But because we are talking about biology, and biology has a lot of variation to begin with, we are always going to be using the value for p equals 0.05. So we locate that column at the top. It's the second column, p equals 0.05, and we move down that column until we reach the row for 28 degrees of freedom. Okay, can you see that with the red circles? And the black arrows are going to take us to the one value that we are looking for, and this value is 2.05. Okay, that is the critical value that we are looking at in this table. Okay, so we write down this critical value, 2.05. Okay, now our critical value is 2.05 and our calculated t value is 1.62. So we can see that the value of t is smaller than the critical value. 1.62 is less than 2.05. When this happens, when the value of t is below the critical value, we accept the null hypothesis. That means that there is no significant difference between the height of students in samples from DC1 and D8. Okay, we accept the null hypothesis that there is no significant difference. Okay, if the value of t is bigger than the critical value, then we have to reject the hypothesis. Okay? Let's look at another worked example. Here we have the table on the right-hand side, and you can see the degrees of freedom are from 1 to 30, and then the values of p are on the top, so that we can look at the corresponding columns. Okay. In this worked example, a researcher measured the wingspans. That means how much the bird measures from one tip of the wing to the other tip of the wing of 12 red-throat and 13 broad-billed hummingbirds. Okay, again with the example of the hummingbirds. Our null hypothesis, there is no significant difference. Okay, our degrees of freedom in this case, how many samples do we have from each population? We have 12 red-throat hummingbirds and 13 broad-billed hummingbirds. So our degrees of freedom would be 12 plus 13 minus 2 would give us a 23 degrees of freedom. Remember that the value for p is always 0.05, and if we look under the correct column, 0.05, we go down until we find the row for 23 degrees of freedom. Therefore, the critical value is 1.714. Okay, so what does this mean? T was calculated, this was given for you in the example, yeah, T was calculated as 2.15, therefore 2.15 is bigger than 1.714, so because T is bigger than the critical value, we have to reject the null hypothesis. Therefore, there is a significant difference between red-throats and broad-bills in terms of wingspan. One group is bigger than the other group, has a bigger wingspan than the other group. They are definitely different. Okay? Why do we reject the null hypothesis if t is bigger than the critical value? If the calculated value for t is greater than the critical value, we reject the null hypothesis. This is because as t increases, the bigger the value for t, we become more confident that the results are real and they are not due to chance, like sampling coincidences that we talked about before. Also notice in this table that as the value of t increases, the value of p decreases. Okay, if we move from the circled 1.714 that was our result, if we move towards the right, we can see that the values for the critical values are going to be bigger, but if we look at the top, the confidence, the value for p is going to be smaller. And the smaller the p-value, the more confident we are. Okay? So if t is less than the critical value, we are less confident that the difference between the means is significant. This corresponds with an increasing value of p. Okay? All right, so in your IB exam, you may be given a value for t and a critical value table, and you may be asked to determine whether two sets of data are significantly different. Now, use the question that is given to you to determine the degrees of freedom, the confidence limits, and compare the calculated value of t to the table provided. So, for example, this is an example question, okay, that was taken from an IB exam. A student measures 16 snail shells on the south side of an island and 15 on the north. She calculates t as 2.02 and chooses a confidence limit of 95%. That means the value of p is 0.05. Are her results significantly different? Again, the null hypothesis is there is no significant difference. Okay? Now, I'm going to give you a few minutes to work this out by yourselves. Okay, remember that the degrees of freedom is the total number of samples minus 2. Remember that she measures 16 snails on one side and 15 snails on the other side. So that would be 16 plus 15 minus 2. Okay? The value for P is given to you in the question, 0.05. And look it up on the table. And what is the critical value? Have you found it? Okay, so in this case, the critical value is 1.699. I know it's hard to see, but that's the critical value. Therefore, you can see in the question, the value for t is 1.61 and the critical value is 1.69. Therefore, t is smaller than the critical value, and that means that you have to accept the null hypothesis. Therefore, your answer should be there is no significant difference between north and south side snails in terms of shell size. Okay? Okay, so the next step is to talk about correlation and causation. Okay? Correlation can suggest relationships between sets of data. That means, for example, using the hummingbirds again, you can have the bill length, how long is their beak, okay? And you can have the weight, how much does each of these birds weigh? And you can have the data there, you can plot this data, and you can see that there is a correlation of this data, okay? In this set of data, there is a strong positive correlation between bill length and body weight. That means that when you look at this graph, you can say things like the bigger the bill length, the higher the body weight. Okay? These are other examples of correlations. If your graph looks like the top left graph, then you can say that there is a strong positive correlation. Okay? The more of X, then the more of Y. Okay? Another example is the second one in the middle. You have a strong negative correlation because you can clearly see that the more of X, the less of Y. Okay? In the third graph over there, you can see all the data points are everywhere. In this case, you can say that there is no correlation because it doesn't show you a trend. It doesn't show you anything that could be going on in there. And finally, in the extreme right graph, the fourth graph from the left, it's called that is on the right, it says a weak positive correlation. Okay, that means, okay, yeah, well, you can kind of see that the more of X, the more of Y, but it's very weak because the data points are kind of scattered. So in some cases is the more of X, the more of Y, but in other cases is not so clear. Okay? How strong is a correlation can be calculated using Excel. We are going to learn how to calculate that by using a best fit line that is going to be giving us the equation for the line, and this equation is going to give us the number of correlation. Okay, so in this case we can see in the bottom three graphs examples of correlation. Okay, when there is a strong correlation, the number is closer to 1. When there is weak or no correlation, the number is closer to 0. Okay, so this is also something that we are going to be practicing using Excel in the classroom, hopefully with the data that you get from the labs. Okay, now one thing is to be able to say that there is a correlation between data. Okay, however, correlations do not prove causality. That means just because two variables are correlated does not mean that one variable causes the other variable. Okay? This is very important. If a correlation exists, further research is needed to determine whether or not the relationship is causal. You can say the more of X that you have, the more of Y. You have a strong positive correlation, but that does not necessarily mean that X or a higher X causes a higher Y. Okay? When you're doing an investigation, that means an experiment, you should have your independent variables, your dependent variables, and your carefully controlled variables in order to see if there is a correlation. And if there is a correlation, maybe perform some more tests to find a causation. That means to see whether one thing really causes the other. Yeah? Some examples of causal relationships, that means that one thing causes the other thing. In this case, we have two variables, temperature versus enzyme activity. We know that there is a strong correlation between temperature and enzyme activity, and in this case, temperature causes enzyme activity to be higher or lower. Okay? Now, another example of causal relationship, concentration versus rate of diffusion. The higher the concentration, the more diffusion happens. Okay, but one thing causes the other. A carbon dioxide concentration versus the rate of photosynthesis. Okay, how much carbon dioxide you have is going to influence how quickly or slowly photosynthesis happens. Okay, by manipulating the independent variable, we can measure a change in the dependent variable. Okay, we can see that one thing causes another. Now, like we said before, correlation does not imply causation. There are some unusual examples of correlation, but not causation. For example, ice cream sales and the number of shark attacks on swimmers are correlated. Maybe because they both happen in the summer, and people in the summer tend to go to the beach more, and therefore get attacked by sharks is more likely, and so on. But that doesn't mean that one thing causes the other. Doesn't mean that the more ice cream you sell, the more shark attacks there are going to be. Okay, they are strongly correlated. However, one thing does not cause the other. Okay, another example of not causation. Skirt lengths and stock prices are highly correlated. As stock prices go up, skirt lengths get shorter. Okay, clearly they are highly correlated, but one thing does not cause the other. The number of cavities in elementary school children and vocabulary size have a strong positive correlation. Clearly, there is no real interaction between the factors involved, simply a coincidence of the data. Okay? It just happened that the higher of one variable, the higher of the other variable, but that doesn't mean that one causes the other. Once a correlation between two factors has been established from experimental data, it would be necessary to advance the research, to make more experiments, to determine what is the relationship between these two variables. Does one necessarily cause the other? Okay, but just because they are correlated doesn't mean that one causes the other. You need more experiments in order to prove that. Okay, so that is the end of statistics. You're probably clapping and you're probably very happy. If not, then wake up, because we finished statistics, and thank you for paying attention. I know this topic is very dry and there's not a lot that we can actually discuss about, but I promise that the future videos are going to be a little bit more entertaining than this one, because they are going to be a little bit better than this one. Okay? Well, thank you, and until the next video.